It's great to see everyone here. I'm so excited and proud to introduce you to Dr. Deanna Mele. She's a former student of mine. She's taken micro. So you can ask her about micro. She graduated from Fairfield University in 2001. And at that point, if I remember correctly, wasn't really sure what she wanted to do in her life. And so she went on to pursue a master's in molecular and biochemistry at Georgetown University, which she completed in 2002. And I moved on to Tufts for a PhD at the Sackler School of Medicine, biomedical sciences, I believe? Yes, in 2008. I don't have the bio.
Around there.
So for a number of years, Dr. Mele was in biotech, and just recently she had a baby and switched jobs and now is working for a big pharma. So she will share with you her research that focuses on cancer immunotherapy, which is a hot field, and for anyone who is thinking about grad school, this is a great area to pursue because these treatments so far have been proven to be quite effective, and that's really the direction where cancer treatment is going. So Deanna will share with you all of her work, and please help me welcome.
Thanks, Dr. Harriet, for the nice introduction. If you can't hear me, just wave, because I'm kind of soft-spoken. So today I'm going to talk to you about the promise of immunotherapy to treat cancer. As Dr. Harriet mentioned, this is an extremely exciting field right now, and I hope to cover some recent advances and breakthroughs, and where pharmaceutical companies and biotech as well are going and headed. But first, I just want to give you more of a career path, because I remember sitting where you are, what feels like not that long ago, but turns out it was a long time ago, and thinking, I have no idea what I want to do. I knew I liked biology. I took all the classes you guys are taking. Start with Intro to Bio. You're like, okay, this is interesting, really hard, but interesting. And then I took genetics, and I loved it even more. I took microbiome, as Dr. Harriet, and loved it even more. And so every, additional, higher up class I took, I found that I was interested in different things. And, part of what I hope to tell you is it's just as important to figure out what you like as what you don't like. So I was a minor in economics here and a major in bio. Looking back, I would encourage all of you to take a business class, whether it's economics or finance, because no matter what you do, that background will help you. And you have the opportunity here to take that class, so you should do it. I graduated in 01, and sort of, I think my junior year, my junior year, I was able to do some research in Dr. Harriet's lab, because I thought, okay, I'm pre-med, but I really don't want to go to medical school, because I had done that internship at Bridgeport Hospital, and for me, the hospital was just not where I wanted to be, and I just knew that I didn't want to go to medical school, which at that time, and as far as I knew, that was kind of what bio majors did. So I thought, okay, well, that's out. Now what am I going to do? So luckily, I had Dr. Harriet and a few others who are sort of mentors for me, and I was able to do some research in her lab and kind of just get a feel for what it means to work in a lab. Although I will say, at that time, the lab was really hard for me because I had no idea what I was doing. And for those of you who are trying to get into a lab or have done research, if you don't love it right now, it's okay. That is normal. I don't think I loved it until I was in my PhD. And that was because getting there was really hard, and I didn't know what I was doing, and you have to depend on the people above you to help you to learn. So, but I did realize that I was really interested in the science, and I wanted to do more. So when I was graduating from Fairfield, I knew I wanted to go to grad school, so I applied to master's programs and PhD programs. But at that point, I really didn't know what I wanted my PhD in, and I didn't want to commit to 5 or 6 years in a PhD program, because that's a long time if you're not sure what you want to do. So I got into both types of programs, and I decided to go to Georgetown. And I did a master's program there through their school of medicine. It was in the biochemistry department, but it was with a specialization in biotechnology. And this was a really cool program, which I would recommend to all of you if you're interested to look into it. And what it does is it gives you the opportunity to take more advanced biology classes, like cancer biology, virology, whatever it is that you're really interested in, or just a class that you take to figure out what you're interested in. But I also took patent law. I took some biotech classes to learn about the business side of things. So I think it just helped me learn what my options were. I didn't have to go get a PhD if I didn't want to. I could go into business and work for a pharmaceutical company and do consulting, for instance, or I could help them figure out, their portfolio and more business side of things. So there are a lot of options out there. But for me, I loved the science and I loved cancer biology. I just thought it was so interesting, and I'm sure as many of you know people with cancer, and to try to understand it, a lot of people still don't understand it, but I was so interested in it that drove me to apply for my PhD. So I knew at that point that I wanted to do something with cancer research, and I applied mainly to graduate programs that were associated with medical schools, because those tend to have more disease-focused labs. Because if you're interested and you do go get a PhD, it's extremely important to have a good mentor, because they are the ones who are going to help you in your career development, and they're going to help you be happy. That's part of your responsibility, but it's part of theirs too. So if anyone has questions about that, I'm more than happy to talk about that, because that process is a whole thing on its own. But what I didn't know when I was sitting near seat that Dr. Harriet told me was, they pay for you. And I said, what? So if you go get your PhD, they give you a stipend. So it's basically enough money to cover your living costs, and you don't have to take out loans like you do for medical school. So for me, that's amazing. You might have to take out a little bit to make ends meet, but it's nothing like the loans you come out with from medical school. So anyway, but in between that, Georgetown and Tufts, I spent a year at Harvard Medical, and the reason was I just missed the timing for applications and stuff like that, and that helped me a lot, because I worked as a tech basically in a lab at the medical school in the cell biology department, and I had a great mentor there. He was amazing. He was one of the smartest guys I've ever met. MD, PhD at 38, assistant professor at Harvard. And he took the time to mentor me, who was just a tech in his lab for basically a year. If you can find someone like that, that's just really going to help you in your career. So, I went to Tufts, and I spent 5.5 years in my PhD. So my PhD was basically cell biology, which was cancer focused, but I also was in a lab that was in an immunology department. So basically I was an immunologist / cell biologist. And not knowing it back then, that turned out to help me immensely into where I am now. And that will tie back to cancer immunotherapy when I sort of get there. But basically through the whole process, I just followed what I loved and what I was interested in. And everyone has a different career path, and you'll figure yours out, but it's good to know what your options are and just always follow what you like. The other thing that I did that many people don't do is, from my PhD, I went right to industry. So I remember, it took me 5.5 years to get my PhD. I was like, I am not doing a postdoc because it's another 5 to 6 years. You get paid nothing, and there's nothing wrong with doing a postdoc. If you want to go to academia, you want to be a professor, it's great. You can really dive into something really deep and do research, and it can be very impactful. But for me, I knew that I didn't want to go that path. And so people had told me, well, you're never going to get a job in industry. Actually, I got 2 jobs. And I took the one at Constellation Pharmaceuticals, which is a biotech company in Cambridge. And when I joined that company, it was only 36 employees. So basically, that was like my postdoc. Again, I had people there that were mentors for me and helped me in the lab and really explained science to me. It's an epigenetics company, so it basically focuses on how genes are turned on and off and how those processes can be involved in cancer. So I learned a lot while I was there, and I kind of used that as my postdoc. So again, I just want to highlight there are a lot of options, and it's just really up to you. You know what you like, what you're interested in, and I encourage you to follow whatever that might be. And then just about like 7 months ago or so, I just started a new job at AstraZeneca. So there's actually a huge difference between biotech and big pharma. So pharmaceutical companies are like Merck, Bristol Myers Squibb, Pfizer, GSK. So all they're really big companies, and they have tons of usually cancer drugs, but they have other drugs as well. But I'm in the cancer field, and that's sort of what I'm talking to you about today. Whereas biotechs are much smaller, and they are more focused on research and early target ID. And so they're trying to find new targets to then develop drugs against them for cancer or other indications. And what ends up happening is actually big pharma ends up eating up a lot of biotechs. And that's because biotechs in general are very innovative, and they're on the cutting edge of research. So if big pharma sees an opportunity, they'll kind of acquire a small biotech and basically take all their technology and targets and develop drugs for them. So that's just a little background on sort of the differences. And again, I'm happy to answer any questions, and just stop me along the way if any come up. So you may be asking yourselves, what is cancer immunotherapy? Well, it's basically activating your own immune system to fight off cancer cells. It's stimulating your own immune system to work harder and smarter to attack cancer cells. So if you have cancer cells in your body, there are many ways that they can trick your immune system from being detected. So what people are trying to do now is to reactivate your immune system to recognize that foreign tumor and basically kill it. But there is obviously challenges to this to do it effectively without causing toxicity. The most common side effect is autoimmunity, and for those of you who don't know what that is, it's your immune system attacks yourself. So there's a lot of autoimmune diseases like lupus, IBS, Crohn's disease, all those types of things where your immune system, for different reasons, attacks yourself basically, and it causes lots of problems. And so that's one of the biggest side effects to cancer immunotherapy. But we've been making a lot of advances and trying to dial that out. In the last couple of decades, immunotherapy has become an important part of treating some types of cancers. And these are just kind of summarizing from the American Cancer Society. Some of the treatments boost your immune system in a very general way, and others are very specific. So it's a relatively broad field. I'm going to talk more about that second one, about more specifically how you use your immune system, specifically your T cells, to attack cancer cells. And then down there is just a quote from the Cancer Research Institute, basically just saying that there's a huge potential of cancer immunotherapy to offer longer-term protection and with fewer side effects, and to benefit more patients with more cancer types. So this is possible that this could be a much more general treatment than some of the more historical ones. So recent cancer immunotherapy breakthroughs, this just illustrates how hot this topic is right now. So if you do a PubMed search, over 66,000 publications come up. And they're all over the place between high impact journals like Science, Cell, Immunity, all different kinds of journals. And Science just came out with 2 years ago and named it the Breakthrough of the Year. And just a couple months ago, the Journal of Clinical Investigation just had a special issue focusing on immunotherapy. So it's everywhere. I mean, it's really pretty amazing. But those are some papers that, if you're interested, you should go PubMed after this and print some of these out just to get more of a background. Because I do believe it's definitely the next big cancer treatment. I mean, it's already happening. So this is actually not a new idea. So the link between immune function and cancer was first proposed actually 150 years ago when Rudolf Virchow observed the prevalence of immune cells in tumors. And so just to give you some context, I mean, this is in New York City in the 1890s. That's really how long ago it was when Dr. William Coley reported survival of some cancer patients with serious infections. He injected mixtures of dead bacteria directly into inoperable tumors, and he treated hundreds of patients with several degrees of success. So basically, even back then, he was able to show that if he used some way, which in this case was dead bacteria, to activate your immune system, it could fight off the cancer. And in 1908, the New York Times published that in their paper that Dr. Coley was able to cure some patients with cancer. Unfortunately, the practice was abandoned as radiotherapy was progressively embraced. So radiation is still used today, as well as chemotherapy, but these treatments are really a sledgehammer, and they're not specific, so they literally wipe out proliferating cells in your body. So even though they can cause some efficacy, a lot of times there's remission, so people come back. They re-present with the cancer because they weren't able to get all the cells. So, most recently, there's been a lot of focus on cancer cell-targeted therapies, and AstraZeneca, as well as big pharma, really have been focused on this in the past, I'd say 50 years, well, maybe 30. Really trying to target oncogenes and tumor suppressors, which are really what drive tumor growth. But the problem with this is resistance. So there are lots of cancer drugs out there on the market that are effective, and it's great. They work really well, although many times the patient will see efficacy, they'll go into remission, and then the tumor comes back. And a lot of this is resistance. So the tumor is unfortunately so smart that it can overcome whatever this targeted therapy was and come up with new ways to kind of go around this therapy. And so you're constantly having to come up with these tumor-driven therapies. So we need new therapies. So it's been pretty exciting. 100 years, actually, after Dr. Coley sort of made his observations, the FDA finally approved a blocking antibody for CTLA-4 for the treatment of metastatic melanoma. This is the first T-cell specific activating drug that they approved. This was in 2011 from Bristol Myers Squibb for melanoma, and they were able to see an increased survival in 20 to 30% of the patients. Unfortunately, there are some serious side effects associated with this, and most of them are autoimmunity. However, shortly after that approval, both Bristol Myers Squibb and Merck came out with 2 blocking antibodies against PD-1, which is a similar target, where it does the same thing. It's just a different target. It activates T cells to kill off cancer. And BMS was able to show a 30% recovery rate in melanoma patients treated with their drug, and in the Merck trial, they showed 45% recovery in these melanoma patients, which is really, really exciting. Let's see here. And then on the bottom, just trying to get the mouse, which is not, there we go. If you look at the lesions on some of these patients before treatment and you do histology and you look at CD8 T cells, which are cytotoxic T cells, which are the ones that actually kill tumor cells, you see some infiltration there, which is in brown. But after 90 days of treatment with anti-PD-1, you now see a huge recruitment of CD8 T cells, which we believe are responsible for fighting off the tumor cells and causing regression. So then they also, Bristol Myers Squibb did a combination study with CTLA-4 and PD-1, and they saw increased efficacy now with the combination, where they see a 53% recovery rate. But really exciting was that they were able to show at least 80% or more improvement in 41% of the patients. I mean, this was unprecedented. When people saw this data, I think it literally led to this. I mean, everyone talks about immunotherapy, and this is back in 2013, where Morgan Stanley published the next wave in cancer immuno-oncology, and similar articles were published in The Economist, the New York Times, Financial Times. I mean, it wasn't just in the scientific journals that people were excited about this, because people saw this as the next new cancer treatment. So, how do these things work? Well, it's extremely complex, and it requires lots of different cell types, but I'll do my best to try to explain that. So tumor cells evade multiple immune checkpoints. So ideally, what happens in a healthy person is they see a cancer cell, their immune system kills it, it's gone, end of story. And other people, for lots of different reasons, the immune system doesn't see the tumor. It grows. You know how a tumor, it metastasizes, and the tumor cells evade multiple immune checkpoints. But the aim of immunotherapy is to re-empower the immune system against cancer cells. So in a normal immune cell, like a T cell for instance, they're wired to cool down after the job is done. So basically after you have an infection, or once you have an infection, T cells get activated along with dendritic cells and macrophages to fight off the infection. And once the infection is cleared, they now kind of go down, cool down, and they stop proliferating and stop secreting pro-inflammatory cytokines. And that's to prevent autoimmunity and chronic inflammation. So that's, what you would want. But tumor cells are so smart that they induce cooling before they're detected. So they can exhaust T cells by expressing certain molecules that basically turn off T cells, as well as cells of the innate immune system. But for this talk, I'm just going to try to focus on the T cell aspect. And tumors can recruit T regulatory cells to suppress inflammation, and these T reg cells are a certain subset of CD4 cells that suppress inflammation. And so what the tumor does is recruits these cells to kind of turn off the pro-inflammatory cytokines that the body might see as a way to attack. So by doing that, they basically hide from the immune system. So just to get into a little bit more biology around the mechanisms of how PD-L1 and PD-1 and CTLA-4 work. So the tumor, as I mentioned, has lots of different cell types in there. You have tumor cells, you have tumor-associated macrophages or dendritic cells, you have fibroblasts, stromal cells, but basically tumor cells can express PD-L1, which is the ligand for PD-1, and PD-1 is expressed on T cells. So what the tumor does is it tries to shut off the T cells by expressing this PD-L1. And so this mechanism here of PD-L1 binding to PD-1 plays a dominant role in suppression of T cell responses. Like I said, many tumors can upregulate PD-L1 expression, inhibiting anti-tumor T cell responses and avoiding immune surveillance and elimination. So durvalumab, which is an AstraZeneca blocking antibody against PD-L1. So now this is blocking the ligand, but again, it's like the same pathway. It blocks the interaction between PD-L1 and PD-1 to overcome inhibition of T cell activation. It significantly inhibits the growth of human tumors in a xenograft model containing co-implanted human T cells. It's currently in phase 3 of clinical trials for non-small cell lung cancer and phase 1 for various other solid cancers. So a lot of the initial data has been generated in melanoma, but now people are testing in other types of cancers, particularly solid cancers, to see if these things can be efficacious in other cancers.
I just want to make sure I understand this. So the drug PD-1
it's a blocking antibody.
prevents activation of the T regulatory cells?
It's a little tricky in Tregs. It's actually the opposite. Is that right? Yeah, prevents activation of Tregs.
Right.
Yeah. Mm-hmm. So then the Tregs go down and the good T cells go up.
Right.
Yep. And CTLA-4 works in a similar way because again it's one of these blocking antibodies for something that negatively affects T cell proliferation. So let's see here. Okay. So normally a T cell gets activated by an antigen presenting cell, like a dendritic cell, and these express antigen, which is recognized by the T cell receptor. It also needs a co-stimulatory signal to become properly activated. And then once the infection is cleared, T cells start to express CTLA-4, which now tells the T cell to stop. You've done its job, so kind of turn off. But like I said, in tumors, they have a way of activating this before the job's done. And so the mechanism by the blocking antibodies is to prevent signaling through CTLA-4. And so now the T cell can't be turned off. And they're able to go to the tumor and kill tumor cells. Again, there's some toxicity associated, especially with CTLA-4, but now lots of other, there's more research being done on other inhibitory receptors. So PD-1 and CTLA-4 are not the only inhibitory receptors expressed on T cells. These are other inhibitory receptors which people are working on and making blocking antibodies for right now, which are hypothesized to have less toxicity but could be less efficacious. It's unclear. But of course, people are really interested in doing combination studies as well. And then on the other side, you have activating receptors. So it's kind of the opposite. So instead of trying to turn off a brake, you go with things that would activate the T cell. So if you had an antibody for OX40 or GITR, these wouldn't be blocking antibodies, they'd be agonists. So they'd be activating these specifically to activate T cells. So how to balance clinical benefit versus safety. So I showed you some data around the anti-CTLA-4 antibodies, which there is some response, but there's also some toxicity. and then the anti-PD-1 where you have an increased response, a decreased toxicity, and then the combo where you had an increased response but you may have increased toxicity. So I think people are trying to look at additional combinations as well as new targets. So as I kind of mentioned, there are lots of different ways that the tumor evades detection. And so the tumor microenvironment is very immunosuppressive. So tumors can recruit immunosuppressive cells such as tumor-associated macrophages, myeloid-derived suppressive cells, and Tregs, or T regulatory cells, to avoid detection and eradication. So ideally from a therapeutic standpoint, you'd want to decrease those suppressive cells in the tumor microenvironment, as well as some of these factors like TGF-beta, IL-10, which are all immunosuppressive, and they can also target area immunosuppression by limiting nutrient availability. So a lot of times the tumor actually eats up a lot of nutrients that the immune cells would actually need to proliferate and fight off the cancer. And so basically they kind of act as sinks, and they just eat up all the arginine, tryptophan, and or cysteine. So now the T cells basically become anergic because they don't have these nutrients around, and so they kind of can't do what they need to do. So I'm going to take a little step back here and just explain a little bit more about immunology, because I'm sure many of you have not taken immunology yet, and it can be a little confusing with all the different subtypes. So I'll try to just give you a brief summary here. Immune cell differentiation. So you start with a stem cell, and then you either differentiate to a common myeloid precursor or a common lymphoid precursor. If you kind of go down here, then this can differentiate to either a T cell, an NK cell, or a B cell. And what I've sort of been talking to you mostly about today is the T cells and how people are trying to activate them to fight off the cancer. But there's definitely research going on in NK cells and B cells as well to try to harness those cell types as well. And then, so these are more adaptive immunity, and this is more innate immunity, where up here where you have the common myeloid precursor, and these can differentiate into granulocytic or monocytic cells. So the granulocytes are sort of up here. The monocytes can differentiate then into macrophages and dendritic cells. And these are really important in innate immunity, which I'll talk about a little bit in the next slide. So the immune system is composed of the innate and adaptive systems. Innate immunity is the first line of defense against pathogens. The response is the same every time, so there's no memory. It's indispensable for survival. There's a direct effect on pathogens. It phagocytoses the pathogen and then lyses it to get rid of it. It's non-specific, but it can recognize self from foreign pathogens. So ideally, it wouldn't cause autoimmunity because it can recognize a foreign pathogen from a self. The cell types that are involved in innate immunity are monocytes, macrophages, dendritic cells, neutrophils, basophils, and mast cells. And this coordinates the adaptive immune system. So particularly macrophages and dendritic cells, if they can't clear the infection, they have ways of activating the adaptive system, which is mainly T cells and B cells. And they pull these cells in to help get rid of the pathogen. So like I said, B and T cells make up the adaptive immune system. They eliminate pathogens by cytokine secretion and antibody secretion, respectively. They're activated by innate immunity, and they have a very specific response because they recognize the antigen or pathogen that is expressed on the surface of mostly dendritic cells. So it's a very specific response, and they have memory, which I'll talk about in a couple slides. So this is just a little picture of the different cell types that are involved in innate immunity. Sort of the ones that we think about most in immuno-oncology are macrophages and dendritic cells and monocytes, because monocytes are basically undifferentiated, and then depending on growth factors, they can either become macrophages or dendritic cells. Just gonna stick with that. The biological function of innate immunity, like I said, is the first line of defense against infection. They phagocytize and digest invading microorganisms, and then they recruit T cell help if needed. It's very important in tissue homeostasis to clear apoptotic cells to prevent autoimmunity. So in all of you have lots of proliferating and dying cells, so it's important to kind of get rid or clear the apoptotic cells so you don't get autoimmunity. Macrophages can present antigens, but not as well as dendritic cells, to mount an adaptive immune response. So dendritic cells are really what we think of as antigen professional cells. And they're the ones that mostly signal to the T cells expressing the antigen on their cell surface to activate the T cells. And this over here is just showing sort of phagocytosis. If there's a pathogen there, how the macrophage, in this case, kind of takes it up, and then that lyses with that fuses with the lysosome and lyses and rids the pathogen. The adaptive immune system evolves over the lifetime of an individual following pathogenic exposure. So, like I said, it's composed of T and B cells that are both derived from the bone marrow, and they express receptors that can differentiate between self and non-self by recognition of protein epitopes. And like I said, they have protein immunological memory. So normally you have a naive T cell, which means it's never seen antigen or never been activated before. And then when it comes in contact with a dendritic cell expressing an antigen, it undergoes clonal proliferation and fights off the infection. And it also generates very few memory cells, and these are long-lived memory T cells, so that when you see the same pathogen again, instead of going through this process, it now is recognized by the memory T cell and can immediately mount an immune response. So this is, T cells are also relatively complex. There are CD8 T cells and CD4 T cells, and CD4 T cells are called T helper cells, and again, that's because when they come in contact with an antigen presenting cell, and they recognize an antigen, they can differentiate into different T helper lineages, and these are, the nomenclature is just like Th1, Th2, Th17, Treg. I don't know why they didn't do Th1, Th2, Th3, for instance, other than I think it's because these are marked by making IL-17, although I don't know why they didn't call that like IL-3, but maybe there's already an IL-3. So. And Th1s and Th2s have been known for a long time. Th17s, I think were just discovered 15, 20 years ago. So, anyway. But basically what happens is, depending on the cytokines that are present as this naive T cell gets activated, it can differentiate into these different lineages, and they basically fight off different kinds of pathogens. And these are all pro-inflammatory except T regulatory cells, and these are what the tumor sort of increases or promotes because it's anti-inflammatory. and sort of as I mentioned, tumors can trick the immune system by suppressing detection, and there's lots of ways that they can do this. So, like I said, normally like a monocyte would differentiate into either a macrophage or a dendritic cell, but the tumor has ways to make this macrophage now a suppressive macrophage, and a dendritic cell to be a suppressive dendritic cell. I mean, it's really amazing, and I think the field is very up and coming, and people are still trying to figure this all out. But I would say, generally speaking, there are three main areas for therapeutic intervention. Antigen presentation, which is innate immunity, like dendritic cells, the T cell response, which I've sort of told you about with anti-PD-1 and CTLA-4, and then overcoming the tumor microenvironment. So this is just sort of showing the tumor here, and what you'd want to happen is you'd have a dendritic cell that was able to detect the tumor and present it as antigen on its cell surface. Here. And then the dendritic cells would migrate to the lymph nodes, where it would come in contact with the T cell. The T cell would recognize the antigen, be activated, and then home back to the tumor and fight off the cancer cells. But unfortunately, there are places all along this pathway that the tumor sort of has hijacked and prevented each process from happening in sort of different ways. But sort of what I told you about today, mostly with the anti-PD-1, is that those blocking antibodies now increase your T cell activity, particularly your CD8 T cells, and they decrease your T regulatory cells. So it's twofold. It blocks some of the suppression in the tumor microenvironment, and it boosts your T cells. So at AstraZeneca, and definitely every other pharmaceutical company in the world, and probably most biotechs, or lots for sure, are entering this IO space, partly because of the efficacy that's been seen in the clinic, as well as there's just so many targets that are sort of waiting to be discovered in the space, because it's relatively new. The field immunology is not new, but sort of now people are coming back to using it in cancer. So here are sort of how we think about strategy at AZ. I told you today about T cell activation part, where we have blocking antibodies for CTLA-4, PD-1, and some others. Another place would be trying to boost antigen presentation by dendritic cells and boosting the innate immunity. And there's lots of ways to do this. It's just trying to find a way that's druggable. And of course, we all want to overcome the suppressive tumor microenvironment. And ideally, people are trying to figure out how to inhibit MDSCs, because these are myeloid-derived suppressive cells, and there's tons of these in the tumor microenvironment that contribute. But part of that is, well, how do we target those and not the good cells? So there's a lot to learn, and it's just a really exciting field. So I just want to close with just some conclusions. So cancer immunotherapy has the potential to cure or transform cancer into a chronic manageable disease. There are a wide array of cancers that are susceptible to immunotherapy, and kind of the interesting thing about this is that this is regardless of mutation, translocation, and amplification. So historically, in lots of drugs that are out on the market now, they target the tumor cells, and those tumor cells have tons of mutations and translocations that drive proliferation and survival, but this is actually not directly targeting these things. It's actually targeting the person's immune system, so it has the potential to possibly not have some of the resistance mechanisms associated with some of these more specific things. Current advances in excitement are based on antibody therapies, and the use of small molecules against targets could be a transformative advance. So, like I said, every big pharmaceutical company is now trying to look at small molecules because everyone's doing the antibody-based therapies, which are great and exciting, but that field, I think, is pretty crowded. And I think there's lots of targets in the small molecule space that people are just trying to figure out. So with that, I'll take any questions. Yep.
Are you finding out that this could be a universal way of fighting cancer cells?
Yeah.
Or is it cancer specific?
I think it's probably both. And we just don't, the answer is we don't know yet. There are some tumors, or some cancer types that have more lymphocyte infiltration than others. And so the idea is those particular cancers would be more susceptible to therapies like this. One of them is melanoma. Another one is lung cancer. And I think breast cancer has a very strong immune function associated with it, so that could be another one as well. So I know lots of combination studies are in trials right now, but people are trying to figure out how you would select patients. Yep.
That was a great overview. I really never even heard of this field.
Yeah.
It's really awesome.
Good.
Because just in the news and stuff, what I keep hearing about is sequencing particular cancers now that the gene,
Yeah.
sequence cells. So I was just wondering if that might provide new targets for things, or this is really more on the immune cells that are coming to the
Yeah, this
cancer.
is more on the immune cells coming in. Actually, the sequencing, I think.
Because that's what I hear is this is the next
Right,
big,
and I
but
think the
this-
news is a little behind. Which is fine. And I think there is value there. But I think that's more on what
It's gonna
people
be-
used to do. And that's
personalized treatments. kind of
yeah, and
for,
I think those might have a longer way to go
Oh,
because
yeah.
it's super expensive, and I don't know how. I don't even know how you would, unless you can find biomarkers. If you can pick out certain genes that you might be able to look specifically for. And actually, I know a lot of companies doing that. Foundation medicine. So if you're interested in that sort of
Well,
thing.
there's definitely, but there seems like there's so many targets already in the immune system for this. It's just like finding the small molecules is now a huge
Yeah,
deal, like you
and
said, so.
I think there's more targets to be found.
Yeah.
For sure. Yeah.
What are some of the limitations? Because you mentioned like a lot of different targets and pathways that can be shut down. Are you finding that some work better than others, and based on side effects?
Yeah, I think the field is still new, but I'm sure that some will work better than others. And I think part of the difficulty is going to be trying to figure out specificity. So, get kind of targeting the right cell types. So, if you have a target that blocks MDSCs, but it also blocks your T cells, that's not a good thing, probably. Your overall effect might even out, so you might not have any efficacy. So I think, there's still a lot of target identification to be done. I think now we know, okay, these are the cell types. Now let's look at receptors or cytokines or sort of more specific targets. Yep.
So, in the clinical trials, what's the major source of the cellular toxicity that's being seen with the blocking antibodies? So, where's the toxicity at the cellular level coming from? What's the source of it?
I think it's just the T cells are just overactivated and they're just attacking. I think I had it in the slide there, which organs they saw.
Do their enzymes spill out and sort of,
What's that?
do their enzymes spill out and kill off? Is there collateral damage or?
I think a lot of it is like where there's a lot of immune cells, so like in the intestine, for instance.
Okay.
There's a lot of toxicity seen there, and some skin, almost like not eczema, but worse than that. So I think it's just an overactivation of the immune system, so that it just
But they're seeing that toxicity in multiple tissues.
yes, yeah.
So what you're basically doing is you're allowing the immune system to do its job by blocking the blockage of the tumor.
Yeah.
Yeah?
Yes. Right. And so the reason why T cells have these breaks is so that they don't cause this autoimmunity, don't attack these organs. But now you've eliminated that break, and so that's what happens. But they're trying to get around that with dosing,
Yeah.
and limiting the times that they have treatment. And like, this is actually a good slide because at the top, red means like CTLA-4 is the biggest break. So it kind of makes sense that the highest tox has been seen with that. And sort of as you go down to yellow, sort of in the primary literature, these have been shown to be sort of less breaks. I don't know how to. So you probably wouldn't get, and a good example is PD-1. It's not as much of a break, but you have less toxicity. So.
You mentioned ways that the tumor is kind of clever and can evade the immune system.
Yeah.
I just want to make sure I understand. So if there are cancers like leukemias where it's not so much a tumor, it's just, are those cells also bad? You know what I mean? Does it
Yeah.
have to be a tumor to be good at evading the immune system?
Yeah,
Or-
no, those cells are good at doing that too.
Okay.
Right.
It's just
So
different.
they're just more circulating.
Right.
And so they do similar things where they secrete cytokines, for instance, that would suppress your immune system. So it's almost, I think it's more like systemic in that sense,
I
where
see.
they're kind of circulating around.
All right, Andrew.
So if the immune system is like overactivated when you're doing this kind of treatment, does it have like an effect on your microbial community, like in your intestines and stuff like that?
Yeah, probably. I think that's why a lot of the tox has been seen there. There are companies that are trying to utilize the microbiome. I just can't remember what the background was. Sorry. Did you
There's a company in Connecticut, what's the one in Ridgefield?
Boehringer.
Yeah, they have a whole section on the gut.
Yeah.
microbiome.
They have a whole division.
Right, so that's definitely,
yeah, and then some companies are looking into like tumor metabolism. So it's kind of like that nutrient part that I was talking about, where the tumor just tries to eat up all the nutrients to suppress the immune system. So they're trying to find targets there if they can, inhibit those things.
any other student questions? I just have one last question, so the human genome's been sequenced, many genomes now have been sequenced, and so I was wondering, we know of genetic disorders like severely combined immunodeficiency, which can lead to increased risk of cancers. Has the genome provided any other insight as to genes involved in the immunity that could play a role? Like, I'm just wondering, like, some of these targets, I'm just wondering how they were discovered. Like, was it at the day?
I think it was like basic research, like in '99, I think it was CTLA-4 that was cloned, or PD-1. Basically, they were looking at things that activated T cells and inhibited. So it's just, I think it was from a basic.
Because I know, like, people have
Immunology
defects in, like, the complement system, and they have an increased risk of, like, infection.
Right.
I'm just wondering if there are, like, disorders, genetic disorders, where there is an immunodeficiency which leads to an increase.
Oh, probably. I don't know of any examples like off the top of my head, but that sounds like something reasonable.
No, I just don't know. Okay, great questions, everyone. Thank you for coming, and let's...